Hello everyone, I'm Yan-Cong Zhang. It's really a great honor to speak here and talk about our recent work on differential expression analysis in the microbiome community. For a microbiome community, one of the key questions we usually ask is, what are they doing? Which means, what function are there? Metagenomics technology help us dramatically understand the functional potential, but these functions couldn't represent they are truly active. In contrast, metatranscriptomics technology have given us more ability to detect the functional activity in the community. Basically, we can non-selectively extract the whole RNA from the community, do the shotgun sequencing, and profile their features using this metatranscriptomics. Usually, we also have the paired metagenomics data, which means from the same sample, we can also extract the DNA and do the sequencing, get the metagenomics data. This MGX data can support our characterization to better understand the metatranscriptomics profilings. Using this MTX profile, we can do a lot of downstream analysis. Not only can look at their expression profilings for a specific clade or specific functional unit, and we can also deep dive more for the regulation process. Today, I want to focus on the statistical analysis, especially the differential expression analysis using metatranscriptomics data. However, there are still a lot of changes in the statistical analysis using MTX data. MTX is highly inflated by zeros, both from the technical zero and the biological zero. Technical zeros are often due to dynamic range from the interacting with the sequencing depth. Biological zeros are common from the missing species or gene loss events. More generally, MTX is highly distinguished from the single organism RNA-seq because for the transcript abundance in the community, highly depends on the underlying gene copy numbers, which means, for example, in increasing the gene copy numbers, which means the microbe carrying it growth via replication, it will correspondingly increase the transcript abundance in count. This will lead to some observation for us from the RNA levels changes. It could due to both by the regulation or by the underlying relative abundance from the species. To illustrate this correlation information between gene copy numbers and its expressions, we can look at this simple community focused on the gene before expression signatures in different situations. For example, in case 1 and case 2, the expression for B4 have changed only due to its underlying gene copy number change, either because from the species abundance increased or due to the gene loss event. In the case 3, this means in this community, the other species, here species A has increased its abundance, leading to the relative abundance for B4 decreased. So this change, the expression is coming from the composition changes, not due to its downregulation. For the case 4, in this case, the B4's gene copy number and its relative abundance have constant with the baseline, but we observed its expression have increased, representing a true upregulation event. To summarize this, we need to pay much attention to normalize this MTX feature when doing statistical analysis. One way for us to look at these MTX features is try to identify the relative expression. For example, in these two samples, we focused on these orange genes, and we can normalize these MTX features within the species in the community. In theory, MTX data could be an equivalent composition of multiple single organism RNA-seq dataset. If we have paired metagenomics data, we can also do normalization by adjust the RNA abundance for the gene copy number using a ratio or residual to represent the relative expression. Given this context, we have developed 6 models to detect the differential expression using MTX data. The first group is a naive model. Basically, we use the overall expression from the community to detect potential expression differences. The second group is using the within taxon normalization approach. We can purely using the within taxon normalized feature to test the differential expression, or we can add more covariate. For here, for example, using the species RNA abundance to adjusting the species bias. The third group is using the paired DNA to normalize the RNA expression informations. We can either use in the RNA DNA ratio or adjust the RNA abundance by the underlying species abundance or gene copy numbers to get the relative expression and test the differential expression profittings. Currently, we have implicated our ideas into linear models, which could easily help us to use arbitrary covariate. In the future, we might need to extending our ideas in more general models. We also consider the technical zeros influence, because for these technical zeros, they could introduce the superior association leading to false positives. We introduce the 3 modes to pre-filter technical zero. The first way is a lenient filtering. We basically ignore the features if it haven't been seen at the RNA or DNA levels. The second approach is a semi-strict filtering. Basically, we exclude the samples for a given features if it have both DNA and RNA zeros. The third way is a very strict filtering approach, which means we treat all zeros as likely technical or uninformative information, exclude samples for genes if it have either RNA zero or DNA zeros. To test our models, we simulated metagenomics and metatranscriptomics dataset using Spartus-inspired models based on healthy human gut microbiome. In these simulations, we include both 2 differential expression signal, and this signal could be confounded by the underlying microbiome species, gene presence, absence, or sequencing depth influence. Using this simulated data, we detected our model's performance. Here, we use the strict filtering approach, basically filter out all the zeros for this non-dataset, which means they don't have any true spiked in differential expression signal. All these methods report okay false positive rate. For the M1 and the M2, they have confused bug abundance changes for differential expression. We can see they have a higher percentage of the false positive rate. For M3, it has been well controlled the false positive and also maintain a good sensitivity. For the DNA-aware methods, they are roughly equivalent here. Attention we need to pay here is that the strict filtering approaches have been applied to test this simulated data, which will prevent model 1, 2, 3, and 5 from confusing gene loss events for differential expression. Moreover, we also compare the different approaches for filtering zeros and their influence on the model's performance. Like we see in the previous slides, the strict filtering have influenced the different models in different situations. When compared with the other approaches, we found M3 have struggled in the presence of technical zeros and co-gene loss events as a differential expression. M5 have behaved very similarly with M3. Interestingly, we found M6, this covariate model, is more robust to zeros than the ratio models, giving strong confounding, which means M6 could be more proper to applied in a situation which including lots of zeros and lots of confoundings. To test our models in real-world dataset, we applied it in a case study. For here, we are particularly interested in the E. coli proteins. This type of proteins have been previously prioritized by MetaVibula, which is a new tool developed by our lab, detecting the potential bioactivity. We found these Peleus proteins have been highly prioritized with potential bioactivity during inflammation. So we hypothesized their expression could also have shown some increased inflammations. So we applied our models to these Peleus and detect potential expression changes. We found that the covariate models have detected a dramatically increased abundance for this E. coli Peleus expression during the IBD-associated inflammation. Similarly, we found the M3, it detected the similar upregulation trend in sign, but it lacked enough statistical significance. One possible reason is that M3 assumed that gene copy numbers are constant within the same species, but in the community, E. coli is a highly strain variant species. So the M3 may be not very proper to apply in these cases for differential expression testing. Surprisingly, we found the ratio models, it detected significant signals in the opposite direction. This observation is counter to our biological intuition, because for the inflammation associated E. coli would be much more likely to upregulate this Peleus structure. It's possible that for this ratio are due to double composition, which means the relative abundance from RNA divided by the relative abundance by DNA could lead to different dominators, leading to superior association direction. So take-home message is that for the covariate model is more robust to small values, can consistent with our hypothesis, also have similar behavior in both simulated and real-world data. To summarize, we recommend that if you don't have paired metagenomics data, M3 could be a reasonable approach. If you do have paired metagenomics data, both M4 and M6 behave well, but M6 could provide a more robust performance to zero values than the ratio model. So currently, we are using M6 as our default. All these implicated models have been wrapped up into our package, and simulated data also provided in our website. Finally, I'd like to thank all the members from the Hot Harbor Lab. Both Curtis and Eric sponsored this project. Kelsey participated in editing the manuscript. I'd like to thank our grant. All the resources and the tools are open to the public. Thanks very much for your attention. I'm happy to take questions.